So our next speaker is Dr. Wang. He is a clinician scientist. He's both an anesthesiologist and a neuroscientist, and he trained at Columbia and at Johns Hopkins and is now at NYU Medical Center. And he's going to be talking about some of the cool new techniques that people are using to study behavior in animal studies. So the effects of prefrontal optogenetic stimulation on chronic pain.
Okay. All right, thanks for that introduction, and thank the organizers for inviting me to talk about some of our recent work on in animal studies. I understand that this today's audience, and there's a lot of interest in clinical translational understanding of chronic pain, early childhood. Influence on chronic pain. Our work is primarily on animal studies, and I'll talk about why. And I think Frank touched on how animal research can inform mechanism and potentially lead to therapeutic insight clinically. And as a physician, I fully appreciate that aspect of basic science research. And so today I want to talk a little bit about how the use of optogenetics, which is a fairly new technique in neuroscience, primarily in rodent work, how the use of this technique how we can use this technique to probe central pain circuits. And specifically, I'll focus on the role of prefrontal cortex in pain regulation. So I don't need to tell the audience today what pain is, but I do want to focus on the fact that pain does have these multiple dimensions, right? There's the sensory dimension of pain, and there's also the affective as well as cognitive dimension of pain. And therefore, as this kind of multi-dimensional experience, it's really important to understand pain in the brain, and which is where all these different dimensions of pain is manifested. In the afternoon, we're going to hear a number of great talks about, from human imaging studies, about how different brain centers interact, functional connectivity that's involved in processing and modulating the pain experience. And so I won't go into all the neuroanatomy and the circuitry here, but I do want to focus on a couple of things, which is that we've always known that there are a kind of parallel pathway in the brain that dictate the sensory, discriminative aspect of pain and the affective motivational aspect of pain. And I think Frank touched on some of that earlier in his talk. But of course, there's a lot of interaction between these two pathways, right? So you also have regions such as like the somatosensory cortex as two regions that might participate in both aspects of pain. Now, the region we're really interested in is the prefrontal cortex, right? So we're interested in prefrontal cortex because the PFC, in short, is a key cortical region that is important for top-down control, right? What do I mean by that? Well, prefrontal cortex projects to a variety of subcortical regions in the brain, regions such as the hippocampus, nucleus accumbens, amygdala, and its projection to these regions are thought to play a very important role in the regulation of emotional or affective type behaviors. And in fact, I think cognitive behavior therapy has been shown to activate prefrontal projection to a variety of these subcortical regions. So that's the role for PFC in kind of emotional regulation. And of course, pain has a very important emotional aspect. And so, of course, PFC plays a very important role in that. And also we know, even just from animal work dating back to the 70s, that opioid or electrical stimulation of the prefrontal cortex can in fact relieve at least certain type of nose-offensive withdrawal experiences, the kind of experiences or behavior that Frank has alluded to earlier. So because of those, that kind of background on the importance of PFC in emotional regulation and in pain regulation, we've become very interested in its role in pain. And what we really try to understand using animal models is that what is the exact role of PFC in processing and regulating the pain experience, not just the sensory, but also the affective pain experience. And more importantly, what are the pathways, or what are the circuit projections that are involved? In other words, what are the outputs of PFC in the brain that are involved in these pain modulatory experiences? Now, a lot of our work has been informed by human imaging studies. Studies done by some of the afternoon speakers, in fact. But I think and I think what Frank alluded to earlier too, is that what animal work is really good for is that we can easily disturb certain circuitry or molecular machinery that might be involved in the brain, and we can see how the perturbations of these molecular or circuit pathways or mechanisms can influence pain, the pain experience, and then we can draw conclusions from these studies, these perturbation as a result of pharmacologic or as I'll introduce, optogenetic manipulations, and how such manipulations can result in pain phenotype, changes in pain phenotype, and giving us clues about mechanisms of how pain arises and can be regulated. So I want to talk about a little bit what optogenetics is. So this is a technique that was developed roughly 5 or 10 years ago by Carl Desroth and Ed Boyden when they're still at Stanford. Let me see how we can get that back on. There we go. When they're at Stanford, right? So what it does is it takes advantage of these, it's actually a very simple concept. It takes advantage of these light-sensitive proteins, or ion channels, essentially. And they're found throughout nature, from bacteria to man. And in fact, that these channels are the reasons we can see light, we can respond to light, our eyes. And so what happened here is that, for example, one commonly used a class of channel ion-sensitive channels are used in optogenetic approach is channel rhodopsin. Now, channel rhodopsins are these ion channels that when they get when they perceive photon and activation, they're able to open and conduct sodium. Okay. Now, so what happens is light shines on these channels, then sodium goes into the cell. Now, when sodium rushes into the cell, particularly a neuron, it can lead to membrane depolarization, right? And that can actually lead to action potential or localized membrane depolarization. Now, localized membrane depolarization then can lead to neurotransmitter release, as shown here, and then leads to the release of neurotransmitters such as glutamate. Okay, and glutamate is an example of excitatory neurotransmitter. And that can then bind to postsynaptic cell to essentially cause synapse. And synapse is the process when one neuron talks to another neuron. So essentially, optogenetic is an approach that allows us, right, to activate directly a neuron, okay, or group of neurons within the brain region, or it allows us to directly activate a connection between one neuron to another neuron, or a group of neurons to another group of neurons at a particular location in the brain. And you can see how this is a very powerful technique that allows us to really essentially understand what a brain region does in a certain behavioral condition. Okay, and so we're going to use this approach to understand what the role of PFC and its projection to a nucleus accumbens is in pain regulation. Okay, and so what we did here is that we used channel rhodopsin. Okay, we linked it to a reporter gene and packaged it into a virus, and then injected this virus into the prefrontal cortex. We then inserted optic fiber. Okay, and then we can shine light through the optic fiber to then activate these these channel rhodopsin proteins, in turn activate those neurons in the PFC, or it's those neurons that project from PFC to the nucleus accumbens. So let's see what happens. And I have to say, all our research is done in rats. Okay, and let's see what happens when we inject the channelrhodopsin into the prefrontal cortex. You can see that we're able to achieve quite a regionally restricted expression of channelrhodopsin. Okay, as shown here by histologically by the presence of yellow fluorescent protein, which is the reporter gene. And you can see that this, by the way, is the region that Frank was talking about in his talk just earlier, the anterior cingulate cortex. So the prefrontal cortex in rats are divided into the prelimbic and infralimbic. And you can see that our work is primarily touched on the prelimbic region, which roughly correlates with dorsolateral PFC in primates. Okay? And you can see that the protein, these channelrhodopsin proteins are expressed in the neurons of the PFC. And furthermore, we also looked at the expression of these channelrhodopsin in the proteins in the nucleus accumbens. And I'll talk about why we're interested in nucleus accumbens in the second part of our talk. But you can see that, though, when we injected the channelrhodopsin into the prefrontal cortex, some of that protein is being expressed and transported along the axon of a prefrontal neuron that goes all the way to the axon terminal of that prefrontal neuron that makes a synapse onto nucleus accumbens. Okay, so we're also able to see a lot of this expression of the channelrhodopsin at the synapse between the prefrontal neurons and the nucleus accumbens neurons, specifically at the core of nucleus accumbens, because the prelimbic PFC projects primarily to the core of nucleus accumbens. And having established that histologic expression of channelrhodopsin, let's see what happens when we shine light. Okay, and you can see that when we shine light onto a brain slice, and so this is a recording from a brain slice, okay, of a rat, and you can see that every time we shine light, we're able to trigger essentially action. And lasers. You can see that. You can see the action potentials, right? Those spikes, and that's when neurons are firing essentially. And this is an in vivo recording in a rat that's anesthetized. Okay, and again, you can see that every time when we shine light through the optic fiber in the brain of this rat, we're able to trigger action potentials in the PFC. Okay, and that's an extracellular recording in a live rat. And so let's see what happens at the behavior level when we activate the channelrhodopsin that are expressed in the prefrontal cortex. Essentially, what will happen when we activate, using light, activate the prefrontal cortical neurons. And let's see what happens to the chronic pain condition. Now, the chronic pain model we used is a spare nerve injury model. Okay, and it's called spare nerve injury model because we take we dissect out 3 branches of a sciatic nerve, and we cut 2 out of 3 branches, sparing the sural nerve. Okay, so hence the spare nerve injury model. And this model was developed in the early 90s by Cliff Wolf at Harvard. And it really mimics the kind of a neuropathic pain as the result of nerve injury from either trauma or surgery, right? And you can see, just like in patients who have neuropathic pain, there are these classic symptoms of neuropathic pain, which are mechanical and cold allodynia. And allodynia, as you all know, refers to the phenomenon where there is the presence of painful experience in the absence of a painful stimulus. So you can see, in other words, when we did this test, and which is essentially the same way we can do this test in humans, is you can see what's the amount of force, of a kind of a blunt force, that's required to elicit a withdrawal response in a rat. Okay, and you can see those rats that have had SNI surgery, that have had their nerve resected, they have this much lower threshold of a response to mechanical and cold stimuli, indicating that there is this kind of a sensory hypersensitivity that's associated with chronic pain. And let's see what happens when we actually activate the prefrontal cortex and what happens to this sensory hypersensitivity symptoms. And you can see that we're able to completely reverse such hypersensitivity symptoms, as indicated by the fact that in those SNI-treated rats, you can see there's a return to essentially control level of a response to normal mechanical stimulus. And the same way with the cold stimulus as well. So what these experiments are demonstrating is that when we activate the prefrontal cortex, we're able to relieve or reduce significantly the sensory hypersensitivity that is associated with chronic pain. But the question we want to ask then is this effect really specific for chronic pain, right? Does the PFC only activate to relieve certain symptoms that are hallmark features of chronic pain, or does it also regulate acute pain as well? And to answer that question, we then perform the following experiment where we use the Hargreaves Test, which is essentially a heat thermal heat pain test. So what we do is we put a rat over a table, and we'll put a radiant heat source under its foot, and to see how long it takes for it to withdraw its foot, right? So the longer it's able to keep its foot on the table, the less pain it feels. And you can see that we took the SNI-treated rats, but here we tested both the injured and uninjured foot, right? So the uninjured foot should really have no neuropathic pain, right? But you can still experience the acute thermal pain. And you can see that for both paws, right? Both the injured and uninjured paw, when we activated channel rhodopsin in the prefrontal cortex, we're able to relieve significantly this thermal heat response, right? So indicating that optogenetic activation of the PFC is not only able to relieve hypersensitivity that's associated with chronic pain, but it's also able to relieve acute pain response. So far I've talked a lot about the sensory response to pain, right? The withdrawal response. But we all know that pain we care about pain not because we care about the sensory component of pain, right? We care about it because it makes us feel bad, right? And you give us all this kind of psychological, psychiatric conditions that are associated with it. And two of the hallmark features of chronic pain is the aversive quality of pain. Well, that's also a feature of acute pain, obviously, but also depressive symptoms of pain, right? And I think Frank talked about both of these components. And unfortunately for us, we can't actually study both of these affective components or dimensions of pain in animal studies. And Frank and Howard really kind of pioneered the use of condition-place preference, the CPP, to assess the aversive quality of pain or the negative reinforcement of pain relief. So I won't go into all the details, and then he's already talked about that in his previous talk. And I think that's really important, though, because it really gives us a way to assess the central nervous system processing, or specifically the brain processing of pain. And that's a really kind of a major advancement in our field of animal research of pain. So we use the same, essentially the same protocol that was developed by Frank. And what we did here is we took those SNI or SNI-treated rats, the rats that have chronic pain, or rats that have undergone sham surgery, which are just control rats, right? And we placed them in and trained them in one of the two, conditioned them in one of the two chambers. In one of the chamber, they received optogenetic activation of the prefrontal cortex. In the other chamber, they had no treatment. So we wanted to see, well, do they prefer the chamber that's associated with their prefrontal activation? And indeed, that's what they demonstrated. You can see that those SNI-treated rats, the rats that have chronic pain, indeed developed this preference to the chamber that's associated with optogenetic activation of the prefrontal cortex. Whereas those sham-treated rats, those rats that didn't have any neuropathic pain, did not care whether their prefrontal cortex is activated or not. So therefore, prefrontal cortical activation has a specific effect in relieving the aversive quality of pain. Okay. And then next, we also wanted to look at the depressive symptoms of pain because, as you know, in chronic pain patients, there is upwards to anywhere between 50 to 100% of comorbidity between chronic pain and depression, okay? And the depressive symptoms of pain can be easily assessed in animals or in rodents with a sucrose preference test and a forced swim test. And these two tests actually have been used to screen antidepressants such as Prozac. So, and what we did here is we just put two bottles of water. One is water, the other is 1% sucrose, okay, and give it to the rat. And you can see that rats, just like us humans, prefer sweet drinks. So sham-treated rats, a kind of control rat, achieved up to 80% preference for sucrose, right? And acid-treated rats, however, those rats that have chronic pain didn't seem to care anymore. So this is a kind of a symptom of anhedonia. Right? And anhedonia is the hallmark feature of depression. Right? So those chronic pain rats, just like chronic pain patients, have this kind of anhedonic symptoms, the symptom of depression. But let's see what happens when we perform the same test while activating the prefrontal cortex. Right? You can see that we're able to completely relieve this symptoms of anhedonia by elevating sucrose preference to the control level. Okay, when we activate the prefrontal cortex in a rat that has chronic pain. Suggesting, therefore, that activation of the PFC can relieve the depressive symptoms of pain. We then corroborated this data, or confirmed this data, with another depression test called the forced swim test. The forced swim test, unlike a sucrose preference test, is not a test for anhedonia, but it's a test for behavioral despair, or learned helplessness. So what we do here is we put a rat in a water tank, and usually a rat wants to swim out of the tank, right? But sometimes he kind of gives up, right? And when he gives up, it stays afloat in the water tank. He doesn't sink. Now, that floating, the time when it stays afloat or giving up on trying to get out of the tank has been marked as immobility or immobility index. And that immobility index has been used to measure, to use as a kind of a measure for behavioral despair or learned helplessness, because he's learned to not swim out anymore. And the higher the immobility index, the higher those symptoms of learned helplessness or behavioral despair. And you can see that compared with a control rat, a rat that has chronic pain developed a significant increase in immobility index. But again, when we performed optogenetic activation of the prefrontal cortex of that rat, those symptoms of immobility or the symptoms of learned helplessness is completely removed. Again, suggesting that prefrontal activation can reduce depressive symptoms of pain. And this is a slide to show that the effect of light activation in the prefrontal cortex is actually quite specific for pain phenotypes, as it did not change gross locomotion. So, optogenetic activation of PFC can abolish acute nociception, or at least reduce acute nociception and sensory component of chronic pain, and that it can also relieve the affective pain symptoms of chronic pain. But we know that PFC has many output projections, right? Which projection actually is important for mediating these pain relieving effects? And that's the next question we ask. And we took some clue from Tor and Vanya's work that they will talk about in the afternoon, and that is the importance of corticostriatal pathway, or the projection from the PFC to nucleus accumbens. And that pathway has been shown to be important for pain appraisal system and for the chronicity of pain. And I won't go into obviously the details because that will be talked about in this afternoon. But we took clue from those studies, and we wanted to know, well, if the corticostriatal pathway is really important in pain, is it because that pathway is disturbed with the chronic pain to make chronic pain worse, or that pathway is being utilized as a kind of an adaptive, endogenous adaptive system to make it better? And to understand that question, we did the following experiments. So to target the corticostriatal connectivity, or that particular circuit, the projection from the PFC to nucleus accumbens, we injected generalopsin into the prefrontal cortex. And then we waited for those proteins to be expressed at the axon terminal of the prefrontal neurons that project all the way to nucleus accumbens. And instead of inserting an optic fiber in the prefrontal cortex, as we have done in these previous experiments I just shown you, we inserted the optic fiber actually in the nucleus accumbens. And what this allows us to do is that it allows us to specifically target those prefrontal neurons that project to the nucleus accumbens. Okay, so it allows us to target the specific synapse or the specific circuit. And let's see what happens to the pain phenotype. Here are some control experiments to show that when we did this, indeed, we see expression of general rhodopsin in the nucleus accumbens, and that when we shine light, we are indeed able to trigger action potentials here in nucleus accumbens neurons. And let's see what happens when we activate this circuit, right? First, we wanted to see what does it do to acute pain state. And you can see that when we repeated the Hargreaves test, we saw that activation of the corticostriatal circuit, just like the activation of prefrontal neurons, is able to relieve acute nociception, okay, or acute nociceptive response of withdrawal, right? But what's really interesting is we then did the following experiments. We tested the thermal response in the right foot, let's say, okay? And most of the studies we've done in optogenetics is we will activate bilaterally, okay, the nucleus accumbens on both sides. But we also, what we did here is that we just activated the side that's ipsilateral or versus contralateral to the side when we're testing. And you can see that we actually didn't see any difference in terms of pain relief at the peripheral level or at the pole withdrawal level, indicating that that the nucleus accumbens, when we activate it, or specifically when we activate the corticostriatal pathway, the projection from the prefrontal cortex to the nucleus accumbens, it doesn't matter if we're activating the left side or the right side. And then next, let's see what happens when we activate this corticostriatal pathway in the chronic pain condition. Are we able to relieve those hypersensitivity symptoms associated with chronic pain? And indeed, you can see that we're able to significantly reduce the amount of mechanical and choroallodynia, perhaps not to the same effect, a full extent as activating the PFC by itself, but still we're able to reconstitute a significant amount of pain relief properties here. We then did the following experiments to confirm the role of this corticostriatal pathway in descending pain control. And what we did here is that we put the optic fiber back in the PFC, okay, to shine light to activate optogenetically those prefrontal neurons. But now, at the same time, we use cannulas to deliver a synaptic blocker specifically in the nucleus accumbens. So essentially what we're doing here is we are activating the prefrontal cortex without activating the nucleus accumbens. Okay, so we allow prefrontal cortical projection to everywhere else in the brain, but just not to the nucleus accumbens. And let's see what happens to pain relief. You can see when by blocking this synapse from the prefrontal cortex to the nucleus accumbens, we are relieving we block the significant amount of pain relief that can be provided by prefrontal activation, suggesting again that this prefrontal cortex to nucleus accumbens connection is very important for pain relief. And what about those affective pain symptoms, right? So here let's take a look what happens when we activate the corticostriatal circuit and perform the CPP experiment again. You can see, again, when we activate the PFC to accumbens projection, we're able to trigger a CPP, or conditioned place preference. So rats prefer that we activate their prefrontal cortical projection to the accumbens in the pain condition. Okay, suggesting therefore that activation of the circuit can also reduce the aversive quality of pain. And finally, we did the experiment of sucrose preference and forced swim again to see if activation of this corticostriatal circuit can also reduce the depressive symptoms of pain. And indeed, that's what we see here. You can see when we activate the projection from the prefrontal cortex to the nucleus accumbens during the test, during the sucrose preference and forced swim test, we're able to completely relieve the symptoms of anhedonia and the symptoms of behavioral despair. Okay, suggesting therefore that the activation of this projection from the PFC to the accumbens can relieve the depressive symptoms that are commonly associated with chronic pain. So in conclusion, I've shown you one way we can use optogenetics to probe central pain circuits, okay, and that PFC neurons indeed provide important modulation for both sensory and affective responses to pain, and that this pain modulatory effect of PFC activation is, at least in part, mediated by its projection to the nucleus accumbens, and thus this particular circuit from the prefrontal cortex to the nucleus accumbens, this cortical striatal circuit, likely plays a crucial role in the regulation of acute and chronic pain. I'd like to thank the people in my lab who've done the work, particularly Michelle Lee and Toby Manders, who've really kind of pioneered the optogenetic studies here. And I'd like to thank the funding agencies, Fairgrant and GMS and NDS. Thank you.